Yeah, next, so చాప్టర్ నెంబర్ 10 ఇంటర్మీడియట్ సెకండ్ ఇయర్ బాటనీలో మనము పాటలు చేస్తున్నాం ఇవన్నీ. ఇప్పుడు ఎన్నో పార్ట్ నాకు కూడా తెలియదు, బట్ 194 పేజ్ నుంచి స్టార్ట్ చేయాలి. 194 పేజ్ నుంచి. ఓకే. ద సిగ్నిఫికెన్స్ ఆఫ్ సచ్ కాంప్లెక్సిటీస్ ఈజ్ నౌ బీయింగ్ టు బి అండర్స్టూడ్. The significance of such complexity is now being to be understood. The split gene arrangements represent probably an ancient feature of a genome. The presence of introns is reminiscent of antiquity. The presence of introns is reminiscent of antiquity, and the process of splicing represents the dominancy of our world. In the recent times, the understanding of RNA and RNA-dependent process in the living system have assumed more importance. Next, genetic code. Genetic code. During both replication and transcription, a nucleic acid is copied to form another nucleic acid. Hence, these processes are easy to encapsulate on the basis of complementarity. The process of translation requires a transfer of genetic information. From a polar of nucleotides to a polar of amino acids. The process of translation requires a transfer of genetic information from a polar of nucleotides to a polar of amino acids. Neither does any complementarity exist between nucleotides and the amino acids, nor could any be drawn theoretically. There exists ample evidence to support the notion that changes in nucleic acid genetic material was possible for change in amino acid in proteins. This leads to the preposition. This leads to the preposition of the genetic material. This leads to the preposition of the genetic code that could direct the sequence of amino acids during the synthesis of the proteins. If determining the biochemical nature of genetic material and the structure of DNA was very exciting. The preposition and deciphering of genetic code were most challenging. In a very true sense, if required, it required involvement of a scientist from several disciplines and physicist, organic chemist, biochemist and geneticist. It was Gregor Gamow, a physicist, who argued that since there are only 4 bases, and if they have to code for 20 amino acids, the code should constitute a combination of bases. This led to the preposition of genetic code that could direct the sequence of amino acid during synthesis of proteins. If determining the biochemical nature of genetic material and the structure of DNA was very exciting, the preposition and deciphering of genetic code were most challenging. In a very true sense, it required involvement of scientists from several disciplines: physicist, organic chemist, biochemist and geneticist. It was Gregor Gamow, a physicist, who argued that since there are only 4 bases, and if they have to code for 20 amino acids, the code should constitute a combination of bases. He suggested that in order to code for all the 20 amino acids, the code should be made up of 3 nucleotides. This was a very bold proposition because because a pre-mutation and combination of because a pre-mutation and the combination of the 4 power 3 అంటే 4 into 4 into 4 would generate 20, would generate 64 codons. So generating many more codons than required. So generating many more requirements than required. కావాల్సిన దానికంటే ఎక్కువ జెనెటిక్ కోడ్స్ ఫామ్ అవుతాయి అన్నమాట అది. So providing proof that the codon was a triplet. Providing proof that the codon was a triplet was a more daunting task. The chemical method developed by Har, Gobi and Corana. Har, Gobi and Coriana. The chemical method developed by Har, Gobi and Coriana. The chemical method developed by Har, Gobi and Corana was instrumental in synthesis of RNA molecules with defined combination of bases. Homopolymers such as UUU and copolymers such as UUC and CCA. Marshall Nirenberg's Marshall Nirenberg's cell-free system to protein synthesis. Marshall Nirenberg's cell-free system for protein synthesis finally helped the code to be deciphered. The enzyme polynucleotide phosphorylase. The enzyme polynucleotide phosphorylase. The enzyme polynucleotide phosphorylase Okaha enzyme named after Severo Okana. Okay, Okana enzyme named after Severina Oka. Okana enzyme named after Severo Oka was also helpful in polymerizing RNA with a defined sequence in a template independent manner. Was also helped in polymerizing RNA with a defined sequence in a template independent manner అంటే enzymatic synthesis of RNA. Finally, a checker board for genetic code was prepared, which is given in table 10.1. The silent features of the genetic code are as follows. ఏంటివంటే the codon is a triplet, 61 codons of code for amino acids and 3 codons do not code for any amino acid. Hence they function as stopped codons. One codon codes for only one amino acid. One codon codes for only for one amino acid. Hence it is a ambiguous and specific. Some amino acids are coded by more than one codon, hence the code is degenerate. Okay, some amino acids are coded by more than one codon. కొన్ని అమినో యాసిడ్స్ ఒక కోడాన్ కాకుండా ఇంకో 2 కోడాన్స్ కలిపి వస్తే దాన్ని డీజనరేట్ కోడాన్ అంటారన్నమాట. నెక్స్ట్ ద కోడాన్ ఈజ్ రెడ్ ఇన్ mRNA in a continuous fashion. The codon is read in mRNA is a continuous fashion. So there are no punctuations. The code is nearly universal. For example, from bacteria to human. The code is nearly universal. For example, from bacteria to human, UUU would code for phenylalanine. Okay, UUU would code for phenylalanine, PHE. Some exception to this rule has been found in mitochondrial codons and in some protozoans. Some exceptions to this rule have been found. Some exception have been found in some exception to this rule have been found in mitochondrial codon and in protozoans. Okay. AUG has dual functions. It codes for methionine MET and also act as a initiation codon. ఈ AUG అనేది 2 ఫంక్షన్స్ ఉన్నాయి. ఒకటేమో మీ కోడ్స్ మిథనైన్ అమినో యాసిడ్స్ కోడ్గా పనిచేస్తుంది అండ్ ఆల్సో యాక్ట్ యాజ్ ఏ ఇనిషియేటర్ కోడాన్గా యాక్ట్ చేస్తారు అనమాట. అది ఈ ఫాలోయింగ్ ఈజ్ ద సీక్వెన్స్ ఆఫ్ న్యూక్లియోటైడ్స్ ఇన్ mRNA. Predict the sequence of amino acid coded by it. Take the help of the checkboard. Okay, so there are no gaps in the actual arrangement of the nucleotides. There are no gaps in the actual arrangement of the nucleotides. Now try the opposite following in the sequence amino acids. Okay, now try the opposite following in the sequence of amino acids coded by an mRNA. So predict the nucleotide sequence in RNA. Predict the nucleotide sequence in the RNA. Met phi 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 phi. Okay. So do you face any difficulty in predicting the opposite? No. Okay. Can you now correlate this with the two properties of genetic code you have learned? Can you now correlate with these two properties of genetic code you have learned? తర్వాత mutations and genetic code. Mutation genetic code ఏంటిదంటే the relationship between genes and DNA, the relationship between the relationship between the relationship between genes and DNA are best understood by mutation studies. You have studied about mutation and its effect in chapter 9. Effects of large deletion and rearrangement in the segment of DNA or in the chromosome. Effects of large deletion and arrangement in the segment of DNA or in the chromosome are easy to comprehend. It may result in loss or a gain of a gene and so a function. The effect of point mutation will be explained here. The effect of point mutation will be explained here. A classical example of point mutation. A classical example of point mutation is a change of a single base pair. A classical example of point mutation is a change of a single base pair in a gene for beta globin chain. In a gene for beta globin chain in human hemoglobin that results in the change in the amino acid. Okay, that results in the change of amino acid result. So okay, గుర్తుపెట్టుకోండి. A classical example of a point mutation is a change of single base pair in the gene for beta globulin chain in human hemoglobin that results in the change of amino acid residue glucota, glu, glutamate to valine. So it results in a diseased cell condition. It results in a diseased condition called the sickle cell anemia. It results in a diseased condition called the sickle cell anemia. How a change in a single nucleotide in a codon alter the amino acid in a polypeptide chain given in figure 10.14. So effect of point mutation that insert or delete the base. ఓకే. దాన్ని అంటే కొత్తది కలపడం గానీ, డిలీట్ చేయడం గానీ జరుగుతుందన్నమాట బేస్‌లో. ఓకే. సో, ఎఫెక్ట్ ఆఫ్ పాయింట్ మ్యుటేషన్ దట్ ఇన్సర్ట్ ఏ బేస్ ఆర్ డిలీట్ ఏ బేస్ ఇన్ ఏ స్ట్రక్చరల్ జీన్ దట్ కెన్ బి బెటర్ అండర్‌స్టూడ్ బై ఫాలోయింగ్ ఏ సింపుల్ ఎగ్జాంపుల్. Consider a statement that is made up of following words, each having 3 letters like a genetic code. Each having 3 letters like a genetic code. Ram has red cap. So if we insert a letter B in between HAS has and LED red, the arrangement of statement would be read as follows. Okay. Ram has రెడ్ కి బదులు పైన చూడండి BRE బ్రీ అండ్ క్యాప్ బదులు DAC DCA ఉంది, తర్వాత P ఉందన్నమాట. సిమిలర్లీ if we now insert two letters at same places, say by, it would read Ram has B and EDC and AP. Now we can insert 3 letters together, say big. The statement would read Ram has big red cap. So the same exercise will be repeated by deleting the letter. The same exercise will be repeated by deleting the letters R, E and D one by one and rearrange the statement to make a triplet word. Okay. Ram has EDC AP, Ram has DCAP and Ram has cap. The conclusion from the above exercise is obvious. Okay, obvious. Insertion or deletion of one or more bases. The conclusion from the above exercise is obvious. Insertion or deletion of one or two bases changing insertion or deletion of one or two bases changes the reading frame from point of insertion or a point of deletion. Okay, insertion of one base or insertion of two base or deletion of one base or deletion of two bases changes the reading frame from the point of insertion or a point of deletion. So insertion or a deletion of three or its multiples bases insert or delete one or multiple codons. Hence one or multiple amino acids. And reading frame remains unaltered from that point onwards. So such mutations are referred to as frame shift. Insertion and deletion mutation. So depending on the change. So this forms the genetic basis of the proof. That codon is triplet and is read in a continuous manner. This forms the genetic basis of the proof that codon is a triplet and is read in a continuous manner. tRNA, the adapter molecule. tRNA, the adapter molecule. From the very beginning of preposition of code. It was clear to Frankie's Crick that there has to be mechanism to read the code and also to link it to the amino acid. And it has a mechanism to read a code and also link it to the amino acid. ఒక లింక్ ఉంది, ఒక కోడ్ ఉందన్నమాట. అంటే కోడ్ చేయడానికి ఒక మెకానిజం ఉంది, అమినో యాసిడ్ లింక్ చేయడానికి అండ్ ఆల్సో టు లింక్ చేయడానికి కోడింగ్కి లింక్కి ఒక మెకానిజం ఉందని చెప్తున్నాడు ఇక్కడ ఫ్రెండ్కి. ఓకే, సో బికాజ్ అమినో యాసిడ్స్ హ్యావ్ నో స్ట్రక్చరల్ స్పెషలిస్ట్ to read the code uniquely. Frankie's Crick postulated the presence of an adapter molecule that would one hand read the code and the other hand would bind to specific amino acid. The tRNA, then called the sRNA, soluble RNA, was known before the genetic code was postulated. The tRNA, then called sRNA, soluble RNA, was known before the genetic code was postulated. However, it ruled as an adapter molecule. Was assigned much later. tRNA has an anticodon loop. That has bases complementary to the code and it also has a amino acid acceptor end with which it binds to amino acid. Okay, tRNA has an anticodon loop that has bases complementary to the code and it also has a amino acid acceptor end with which it binds to amino acids. Okay, tRNA are specific for each amino acid. Figure 10.15. For initiation, there is another specific tRNA that is referred to as the initiator tRNA. So there are no tRNAs for stop the codons. There are no tRNAs for stop codons. Okay, in figure 10.15, secondary structure of tRNA has been depicted that looks like a clover leaf. The secondary structure of the tRNA has been depicted that looks like a clover leaf. In actual structure of tRNA, the tRNA is a compact molecule. Which looks like an inverted L. Okay, next translation గురించి మాట్లాడదాం. సో ట్రాన్స్లేషన్ అంటే ట్రాన్స్లేషన్ refers to the process of polymerization of amino acid to form a polypeptide, figure 10.16. The order The order and sequence of amino acids are defined by a sequence of bases in the mRNA. The order and sequence of amino acids are defined by the sequence of bases in the mRNA. The amino acids are joined by a the amino acids are the amino acids are joined by a bond which is known as the peptide bond. Formation of peptide bond requires energy. Therefore, in the first phase itself, amino acids are activated in the presence of ATP and linked to their cognate tRNA a process commonly called as the charging of tRNA or amino acylation of tRNA, charging tRNA or amino acylation tRNA to be specific. So if two such charged tRNAs are brought close enough, the formation of peptide bond between them would be favored energetically. The formation of peptide bond between them would be favored energetically. The presence of a catalytic would enhance the rate of peptide bond formation. Okay, the presence of a catalyst would enhance the rate of peptide bond formation. The presence of a catalyst would enhance the rate of peptide bond formation. The cellular factory responsible for synthesizing the proteins in the ribosome. The cellular factory responsible for synthesizing the proteins is the ribosome. The ribosome consisting of structural RNAs. And about 80 different proteins. In its inactive state, it exists as two subunits, a large subunit and a smaller subunit. When the smaller subunit encounters an mRNA, the process of translation of mRNA to proteins begins. The process of translation of the mRNA to proteins begins. So there are two sites in the large subunit for subsequent amino acid to bind to and thus to be close enough to each other for the formation of peptide bond. The ribosome also act as a catalyst in bacteria. The ribosome also act as a catalyst for the formation of peptide bond. గుర్తుపెట్టుకోండి ఇక్కడ ఏ రైబోజోమ్ బ్యాక్టీరియా అంటే 23S rRNA in bacteria is the enzyme 23S rRNA in bacteria is the enzyme ribozyme. Okay, ribozyme for the formation of peptide bond. Okay, a translation unit in mRNA. A translational unit in mRNA is the sequence of RNA that is flanked by the start codon AUG and stopped codon and codes for polypeptide. చూడండి ఇక్కడ ఏ ట్రాన్స్లేషన్ యూనిట్ అనేది ఒక ట్రాన్స్లేషన్ యూనిట్ ఒక పదం చూడండి. ఏ ట్రాన్స్లేషన్ యూనిట్ ఇన్ mRNA is a sequence of RNA that is flanked by the starting codon AUG and stop codon and codes for the polypeptide. ఈ మూడుతో జరుగుతుంది, ఫ్లాంక్ జరుగుతుంది. An mRNA also has some additional sequences that are not translated and are not referred to as untranslated regions. The untranslated regions UTR. The UTRs are present at both 5' end before start codon. The UTRs are present at both 5' end and at 3' end before start codon and after stop codon. Before start codon, after stop codon. చూడండి, స్టార్ట్ కోడానికి ముందు, స్టాప్ కోడానికి తర్వాత ఓకే ఉంటాయన్నమాట ఇవి. సో UTRs are present. Okay, 5 end and at 9 end, 3' end. So they are required for efficient translation process. Of the process. For initiation, the ribosomal For initiation, the ribosome binds to the mRNA at the start codon AUG that is recognized only by the initiator RNA. That is recognized only by the initiator tRNA. The ribosome proceeds to the elongation phase of the protein synthesis. The ribosome proceeds to the elongation phase of the protein synthesis. So during this stage, complex X composed of an amino acid linked to tRNA sequentially bind to the appropriate codon in mRNA by forming complementary base pairs with the tRNA anticodon. So the ribosome moves from codon to codon along the mRNA. The ribosomes moves from codon to codon along the mRNA. Amino acids are added one by one. Amino acids are added one by one, translated into polypeptide sequences, and dictated by DNA, translated into polypeptide sequences dictated by DNA and represented by mRNA. At the end, a release factor binds to the stop codon, terminating the translation and releasing the complex polypeptide from the ribosome. And releases the complex polypeptide from the ribosome. And releasing the complete polypeptide from the ribosome. Next to translation. Translation refers to the process of polymerization of amino acid. Translation refers to the process of polymerization of amino acid to form a polypeptide, figure 10.16. The order and sequence of amino acids are defined by the sequence of bases in the mRNA. The amino acids are joined by a bond which is known as the peptide bond. Formation of peptide bond requires energy. ఇదంతా నేను చదివేసిన ఇదంతా చదివేసినాను. ఎక్కడి నుంచి అంటే ఇక్కడ తప్పు చదివినాను. ఇక్కడ 199 పేజీ నుంచి చదవాలా. ఓకే, రైట్. సో 199 పేజీలో నుంచి మనము రెగ్యులేషన్ ఆఫ్ జీన్ ఎక్స్ప్రెషన్ నుంచి చదవాలి. అది గురించి స్టార్ట్ చేస్తాను. సో రెగ్యులేషన్ ఆఫ్ జీన్ ఎక్స్ప్రెషన్ ఏంటంటే, రెగ్యులేషన్ ఆఫ్ జీన్ ఎక్స్ప్రెషన్ refers to a very wide sequence of events that may occur at various levels. Considering that gene expression resulting in the formation of polypeptide. Results in the formation of polypeptide. So it can be regulated at several levels. It can be regulated at several levels. In eukaryotes, the regulation could be exerted. At transcriptional level, at processing level, and transport of mRNA from the nucleus to cytoplasm level. తర్వాత ట్రాన్స్లేషన్ లెవెల్ గుర్తుపెట్టుకోండి, 3 లెవెల్స్లో అవుతుంది. ఇన్ యూకారియోటిక్స్ ద రెగ్యులేషన్ కుడ్ బి ఎగ్జర్టెడ్ ఎట్ 4 లెవెల్స్లో జరుగుతుంది. అంటే ఆగిపోతుందేమో. ఏదంటే ట్రాన్స్క్రిప్షన్ లెవెల్ అంటే ఫార్మేషన్ ఆఫ్ ప్రైమరీ ట్రాన్స్క్రిప్ట్. తర్వాత ప్రాసెసింగ్ లెవెల్, దీన్నే రెగ్యులేషన్ ఆఫ్ ది స్ప్లైసింగ్ లెవెల్ అంటారు. తర్వాత మూడవది ట్రాన్స్పోర్ట్ ఆఫ్ mRNA ఫ్రమ్ ద న్యూక్లియస్ టు సైటోప్లాజం. తర్వాత ఫోర్త్ ఏమో ట్రాన్స్లేషన్ లెవెల్. అది ద జీన్స్ ఇన్ ఎ సెల్ ఆర్ ఎక్స్ప్రెస్డ్ టు పర్ఫార్మ్ ఏ పర్టికులర్ ఫంక్షన్ ఆర్ ఏ సెట్ ఆఫ్ ఫంక్షన్స్. ఫర్ ఎగ్జాంపుల్, an enzyme called beta galactosidase. Okay, for example, an enzyme called the beta galactosidase is synthesized by E. coli. It is used to catalyze the hydrolysis of a disaccharide. Lactose into galactose and glucose. Bacteria use them as a source of energy. Bacteria use them as a source of energy. If bacteria, hence, if the bacteria do not have lactose around them to be utilized for energy source, they would no longer require the synthesis of enzyme beta galactosidase. Therefore, in simple terms, it is the metabolic and physiological or environmental conditions that regulate the expression of genes. In simple terms, it is the metabolic, physiological, or environmental conditions that regulate the expression of the genes. The development and differentiation of embryo into adult organisms is also a result of the coordinated regulation of expression of several set of enzymes. The development and differentiation of embryo into adult organisms is also a result of the coordinated regulation of expression of is also a result of the coordinated regulation of expression of several set of genes. Coordinated regulation of expression of the several set of genes. Okay, right. In prokaryotes, control of the rate of transcriptional initiation. In prokaryotes, control of the rate of transcriptional initiation is the predominant target site for controlling of gene expression. In prokaryotics, control of the rate of transcription initiation is a predominant target site for control of gene expression. In a transcription unit, the activity of RNA polymerase at a given promoter is in turn regulated by interaction with accessory proteins, which affects the ability to recognize the start sites. These regulator proteins can act both positively or negatively. These regulatory proteins act both positively అంటే activators and negatively repressors. The accessibility of promoter region of prokaryotic DNA. The accessibility of the promoter region of prokaryotic DNA is in many cases regulated by the interaction of proteins with sequence termed the operator. Okay, operator. The operator region is adjacent to the promoter elements. The operator region is adjacent to the promoter elements in most operand. In most cases, the sequence of operator bind to the repressor protein. చూడండి ఇక్కడ the operator region is adjacent to the promoter elements in most operands, and in most cases the sequence of operator bind to the repressor protein. Each operand has its specific operator and specific repressor. గుర్తుపెట్టుకోండి, each operand has its specific operator and specific repressor. For example, lac operator is present only in the lac operon and it interacts specially with the lac repressor only. Okay, అది lac operon. The elucidation of the lac operon was also a result of the lac operon was also a result of close association between a genetist. Elucidation of the lac operon was also a result of a close association between the genetist Francis Jacob and biochemist Jacques Monod. Okay, Francis Jacob and biochemist Jacques Monod. These scientists చూడండి ఇక్కడ జెనెటిక్ అనే సైంటిస్ట్ ఏంది ఫ్రాంకోసిస్ జాకోబ్ అంటారు, బయోకెమిస్ట్ని జాక్వి మోనోబ్ అంటారు. So these scientists were the first These scientists were the first to elucidate a transcriptional regulating system. These scientists were the first to elucidate a transcriptionally regulating system. In lac operon, here lac refers to lactose, a polycystonic structural gene. A polycystonic structural gene is regulated by a common promoter and regulatory genes. Such an arrangement is very common in bacteria and is referred to as operon. To name a few such examples, lac operon and trioperon and ara operon and his operon, val operon, etc. Lac operon, trip operon, ara operon and his operon, val operon, etc. The lac operon consisting of one regulatory gene. The lac operon consisting of one regulatory gene. The I gene here the term I does not refer to induce. Okay, I gene here the term in here. Okay, the lac operon consisting of one regulatory gene. Okay, the I gene here the term I does not refer to inducer, so rather it is derived from the word inhibitor. Okay, the promoter P and operator O, and three structural genes Z, Y, A. The I gene codes for the repressor of the lac operon, and the Z gene codes for beta galactosidase B-gal enzyme, which is primarily responsible for the hydrolysis of the disaccharide lactose into its monomeric units galactose and glucose. Okay, the Y gene code for an enzyme primerages. The Y gene codes for enzyme primerages, which increases permeability of the permeability of the cell to beta galactosides. So, which increase the permeability of the cell to beta galactosides. The A gene encodes a trans-acetylase enzyme. The A gene encodes a trans-acetylase enzyme. Hence, all the three genes products in lac operon are required for the metabolism of lactose. Hence, all the three gene products in lac operon are required for the metabolism of lactose, and in most other operon as well. In most other operands are as well. In most other operands as well. ఓకే, ఇన్ మోస్ట్ అదర్ ఓపరెంట్స్ యాజ్ వెల్ అంటే మిగతా ఎక్కువ శాతం ఓపరెంట్స్ కూడా అలాగే ఉన్నాయని చెప్తున్నాడు. ఓకే, ఇన్ మోస్ట్ అదర్ ఓపరెంట్స్ యాజ్ వెల్ ద జీన్స్ ప్రజెంట్ ఇన్ ద ఓపరాన్ ఆర్ నీడెడ్. The genes present in the operon are needed together to function in the same or related metabolic pathway figure 10.17. Lactose is a substrate for the enzyme beta galactosidase. Lactose is a substrate for the enzyme beta galactosidase, and it regulates the switching on and off of the operon. Hence, it is termed as inducer. In the absence of a preferred carbon source. Such as glucose. Okay, in the absence of a preferred carbon source such as glucose, if lactose is provided in the growth medium of the bacteria, it is transported into the cells. Okay, it is transported into the cells through the action of permease. Remember, a very low level of expression of lacto-operon has to be present in the cell all the time. So remember, a very low level of expression of the lac operon has to be present in the cell all the time. Otherwise, lactose cannot otherwise lactose cannot enter the cells. Otherwise lactose cannot enter the cells. The lactose then induces the operon in the following manner. The lactose then induces the operon in the following manner. ఎలా అంటే the repressor of the operon is synthesized all the time, hence said to be consequently from the I gene. The repressor of the operon is synthesized all the time from the I gene. The repressor protein binds to the operator region of the operon and prevents the RNA polymerase from transcribing the operon. In the absence of in the presence of an inducer such as lactose or in the presence of an inducer గుర్తుపెట్టుకో in the presence of an inducer such as lactose or allolactose the repressor is inactivated by interaction. The repressor is inactivated by interaction with the inducer. Okay, this allows RNA polymerases to access the promoter and transcription proceeds. This allows RNA polymerase to access the promoter and transcription proceeds. Figure 10. And the figure 10.17. Okay, essentially the regulation of lac-operon can also be visualized as a regulation of enzyme synthesis. Okay, essentially the regulation of lac-operon can also be visualized as a regulation of enzyme synthesis by its substrate. So remember glucose or galactose cannot act as an inducer for lac-operon. Can you think for how long the lac-operon would be expressed in the presence of lactose in the presence of lactose? Okay, the regulation of lac-operon by repressor is referred to as negative regulation. The regulation of lac-operon by repressor is referred to as a negative regulation. The lac-operon is under the control of positive regulation as well, but it is beyond the scope of discussion at this level. Okay, summary చూస్తే nucleic acids are very long polymers of nucleotides. While DNA stores genetic information, RNA mostly helps in transfer and expression of the information. ఇంతకుముందు చెప్పుకున్నాం డిఎన్ఏ అనేది జెనెటిక్ ఇన్ఫర్మేషన్ స్టోర్ చేస్తుంది, ఆర్ఎన్ఏ మాత్రం ఎక్కువ శాతం ట్రాన్స్ఫర్ చేస్తుంది, మరి ఎక్స్ప్రెస్ చేస్తుంది ఇన్ఫర్మేషన్ అని తెలుసుకున్నాం అనమాట. ఓకే, though DNA and RNA both function as a genetic material, DNA is chemically and structurally more stable and is a better genetic material. However, RNA was the first to evolve and DNA was derived from RNA. The hallmark of the double-stranded helical structure of DNA is the hydrogen bonding. The hallmark of the double-stranded helical structure of DNA is the hydrogen bonding between the bases from opposite strands. The rule is that adenine pairs with thymine through 2 hydrogen bonds and guanine with cytosine through 3 hydrogen bonds. This makes one strand complementary to another. The DNA replicates semi-conservatively. The process is guarded by the complementary H bonding. A segment of DNA that encodes for RNA may in simplistic term be referred to as gene. Okay, during transcription also one of the strands of DNA act as a template. Okay, during transcription also one of the strands of DNA act as a template to direct the synthesis of complementary RNA. In bacteria, the transcripted mRNA is functional, hence can directly be translated. In eukaryotes, the gene is split. The coding sequences Exons are interrupted by the non-coding sequences introns. Introns are removed and exons are joined together. Introns are removed and exons are joined to produce the functional RNA by the process called the splicing. Okay, splicing. Splicing. Okay, splicing అంటే దాన్ని. The messenger RNA contains the base sequences that are read in a combination of three to code for amino acid. Okay, అంటే ఇక్కడ టు మేక్ ట్రిప్లెట్ జెనెటిక్ కోడ్ మూడు ఉంటే మనకు జెనెటిక్ కోడ్ మూడు ఉంటే దాన్ని ట్రిప్లెట్ అంటారు అనమాట. అది the genetic code is read again on the principle of complementarity by tRNA that acts as a adapter molecule. There are specific tRNAs for every amino acid. There are specific tRNAs for every amino acid. The tRNA binds to the specific amino acid at one end and pairs through hydrogen bonding with codes on mRNA through its anticodons. The site of translation protein synthesis is ribosomes. That is the site of translation అంటే దాన్నే the site of photosynthesis అంటే ribosomes, which bind to mRNA and provide a platform for joining of amino acids. One of the rRNA act as a catalyst for peptide bond formation, which is an example of the RNA as an enzyme ribosome. Okay, ribosome. Translation is the process that evolved around RNA. Translation is a process that has evolved around RNA, indicating that life began around RNA. Since transcription and translation are energetically very expensive process, so these have to be tightly regulated. Regulation of transcription is the primary step for regulation of the gene expression. In bacteria, more than one gene is arranged together and regulated in units called the operon. Lac operon is the prototype operon in bacteria which codes for genes responsible for metabolism of lactose. The operon is regulated by the amount of lactose in the medium where the bacteria are grown. Therefore, this regulation can also be viewed as a regulation of enzyme synthesis by its substrate. So therefore, this regulation can also be viewed as a regulation of enzyme synthesis by its substrate అన్నమాట. తర్వాత ఏమంటారు అంటే నెక్స్ట్ గ్లోసరీకి వస్తే యాంటీకోడాన్ అంటే a sequence of 3 adjacent nucleotide in tRNA that is complementary to the codon in messenger RNA which specifies the amino acid is called anticodon. Complementarity అంటే it is the property shared by two nitrogenous bases or two polynucleotide strands which when positioned opposite to each other each other can pair by forming hydrogen bonds between AT or U and between G and C. Okay, AT or U, A అనేది T తోనైనా కానీ లేకుంటే A అనేది U తోనైనా కానీ లేకుంటే G కి C కి మధ్య అనమాట. అలా double helix, it is the second order structure formed by two complementary deoxy polynucleotide chains twisted around each other in mutual opposite orientation and held together by nitrogen bonds. Gene regulation. So gene regulation, it is the process of turning genes on and off so that appropriate genes are expressed at proper times and places. Is called gene regulation. Genetic code ante the set of all possible 3 nucleotide combinations in DNA or mRNA that combine the specific amino acid sequence in a polypeptide chain. In a polypeptide chain is called genetic code. Genomics, it is the field of study dealing with the determination of the nucleotide sequences of the genome and its analysis using complete program and the analysis using computer program is called genomics. Histones. Histones are the chief protein components of chromatin in eukaryotic cells. Histones are the chief protein components of chromatin in eukaryotic cells, acting as spool around which DNA winds and play a role in the gene regulation. HRNA, heterogeneous nuclear RNA. Heterogeneous nuclear RNA found in the nucleus which becomes mRNA after undergoing processing or maturation is called heterogeneous nuclear RNA. mRNA, it is the form of RNA that carries information from DNA to ribosomes and function as the template for protein synthesis. Negative regulation. The method of regulation of operon or a gene expression where repressor protein binds to the operator site, preventing the expression of structural genes is called the negative regulation. Nucleosome. It is the basic unit of DNA packing. Nucleosome అంటే it is the basic unit of DNA packing into chromatin and consisting of a segment of DNA wound around a protein core of 8 histone molecules. So 200 base pairs అనమాట, అది is called nucleosome. Operon అంటే operon. So it is a group of closely packed structural genes and regulatory elements DNA sequences such as a regulator, a promoter and a operator which functions in a coordinated manner is called operon. Operator. It is a segment of DNA to which the product of regulatory gene protein bond. Operator ante it is a segment of DNA to which the product of regulatory gene protein binds. In the lac operon, it is a segment between the promoter and the structural genes of the operon is called operator. Promoter, it is a region of DNA where RNA polymerase bind and initiate the transcription is called the promoter. Replication fork, it is a site on a DNA double helix where both unwinding of helix and synthesis of the daughter molecule occurs is called replication fork. Next, structural gene. It codes for mRNA that determines the amino acid sequence in a protein or in a polypeptide chain other than a regulatory protein is called structural gene. snRNA అంటే small nuclear ribonucleic acids found within the eukaryotic nucleus. They are involved in some important processes such as RNA splicing and regulation of transcription factors. తర్వాత tRNA అంటే ట్రాన్స్ఫర్ RNA is an adapter molecule that is used by all living organisms to bridge the genetic code in messenger RNA with the 20 amino acids in protein. tRNAs carry amino acids to ribosomes. tRNAs carry the amino acids to ribosomes where they are linked into proteins. tRNAs carry amino acids to ribosomes where they are linked into proteins. tRNAs carry amino acids to ribosomes where they are linked into proteins. ఈజ్ కాల్డ్ ది ట్రాన్స్ఫర్ ఆర్ఎన్ఏ. అంతే. ఈ చాప్టరు ఇంటర్మీడియట్ సెకండ్ ఇయర్ బాటనీ చాప్టర్ నెంబర్ 10 అయిన మాలిక్యులర్ బయాలజీ అంటే మాలిక్యులర్ బేసిస్ ఆఫ్ ఇన్హెరిటెన్స్ అనే చాప్టర్ కూడా ఇంతటితో సమాప్తం. ఇంకా అంతా అయిపోయింది. ఓకే. సో ఇంటర్మీడియట్ సెకండ్ ఇయర్లో అన్ని చాప్టర్స్ రికార్డ్ చేసేసినట్టే.